Great, good afternoon to all. Yesterday we discussed about the topic what happened to camera? Yesterday we discussed about the hypersensitivity. Means the ability of the immune system to respond appropriately to the antigenic challenge that is termed as hypersensitivity. The hypersensitivity reactions, those were the immune responses that are exaggerated or inappropriate against an antigen or allergen. This inappropriate immune response that can have the deleterious effects that resulting in the significant tissue damage or even death. The immune reactions that releases the inflammatory mediators that result in a increased vascular permeability and recruitment of inflammatory cells to cause the local tissue inflammation. The hypersensitivity describes the tissue damage, the secondary to an exaggerated inflammatory reaction. These reactions may be in response to external antigenic challenges. Those were like pollen grains in hay fever, an appropriate response to a pathogen, or even to an individual's own tissue. So, according to the scientist Gell and Coombs, here the hypersensitivity is of four types. Those were type 1, type 2, type 3, type 4. Type 1 hypersensitivity, this is also called IgE mediated mast cell degranulation. So, in this case, the type 1 the type 1 hypersensitivity, again called as allergic reaction. Here in these reactions, those were rapid, occurring within the minutes after the re-exposure to the antigen. Thus, allergic reactions are also called as immediate hypersensitivity. This immediate type of hypersensitivity that is mediated by the antibodies called IgE antibodies. There are many antigens which can cause allergic hypersensitivity reactions. The common allergic response is probably to the pollen grains. Those were like allergic rhinitis, hay fever. Antigens from some invading organisms that can also give rise to allergic reactions. These include the fungal spores and viruses and worms. So the type 1 hypersensitive response from a normal humoral response is that the plasma cells that secrete IgE, then the IgE antibody that binds with the high affinity to Fc receptors on the mast cells and basophils, So the mast cells and the basophils that is coated by the IgE that are said to be sensitized. These sensitized mast cells and the basophils that synthesize the primary mediators and store in the granules. The second exposure to the same allergens that cross links the membrane bounded IgE on sensitized mast cells and basophils that cause to release of the secondary mediators called degranulation. So the releasing of the uh, active secondary mediators, those like vasoactive amines and leukotrienes and cytokines and histamines, those called the smooth muscle contraction that increased vascular permeability and vasodilation. Here, the atopy means the generation of inappropriate levels of IgE against the external allergens. So here, the generation of inappropriate levels of IgE against the external allergens is called as atopy. For example, allergic conjunctivitis and allergic rhinitis, allergic dermatitis, and foodborne infections, foodborne allergies, and anaphylactic shock and bronchial asthma. These are all the examples for the type 1 hypersensitivity reactions. So when coming to the hypersensitivity type 2 reactions, those were called as antibody dependent cytotoxicity. So these antibody dependent cytotoxicity or cytotoxic reaction or cytolytic reaction and the type 2 hypersensitivity is caused by antibodies that is binding to the cells or the tissues and antigens. Here the IgG and IgM. In case of type 1, IgE is the antibody that binds with the high affinity to the Fc receptors on the mast cells or and basophils. But here in case of type 2, IgG, IgM is responsible for these reactions. So I then called IgG and IgM mediated cytotoxic response against the cell surface and extracellular matrix proteins. The immunoglobulins that involved in this type of reaction that damages the cells by activating the complement system or by phagocytosis mechanism. So there are two different types of antibody mediated mechanisms that are involved in these cytotoxic reactions. So where the involvement of the natural killer cells that killing the target cell by the uh, effect of macrophage or NK cell. In the other hand, IgG, it will fix the complement activation. It will activate the complement where the killing of target cell by complement mediated lysis. So what is complement mediated hypersensitivity means? In this reaction, the antibody reacts with a cell membrane component that leading to the complement fixation. This activates the complement cascade and leading to the lysis of the cell or opsonization. The blood cells that are most commonly affected by this mechanism. The other one is antibody dependent cell mediated cytotoxicity. Here in this case, The antibody dependent cell mediated cytotoxicity reactions involve IgG and IgG FC receptors. This antibody dependent cell mediated cytotoxicity used FC receptors that is expressed on many cells such as natural killer cells, macrophages, eosinophils, neutrophils as a meaning of bringing these cells into contact with the antibody coated target cells. The lysis of these target cells that require contact but does not involve phagocytosis or complement fixation. So, the antibody dependent cell mediated cytotoxicity that lysis involves the releasing of cytoplasmic granules which containing the porphyrin and granzymes that activate the events that leading to apoptosis. For example, erythroblastosis fetalis and autoimmune hemolytic anemia and immune thrombocytopenia and autoimmune neutropenia. These are all the examples for the type 2 hypersensitivity. When coming to the type 3 hypersensitivity, here it is also called as immune complex mediated hypersensitivity reaction. It is mediated by IgM and IgG antibodies that reacts with soluble antigens that forming antigen antibody complexes. The antigen antibody complexes with microbial or auto antigens that results in immune complexes that the deposition in the blood vessels is that condition is called as vasculitis, or in the renal vessels glomeruli of kidneys that leading to glomerulonephritis. So antigen antibody complexes that are maintained as soluble immune complexes in blood by complement proteins like C2 and C4. These soluble immune complexes that bind to complement receptors on RBC that allowing their transport to the spleen, where the complexes are removed and destroyed if the production of immune complexes is greater than the clearance. Their accumulation can leading to a hypersensitivity reaction from complement that triggers a variety of inflammatory processes. For example, the Arthus reaction and serum sickness, the pulmonary diseases that results from inhalation of the bacterial spores. It is also that condition is also called as like farmer's lung or avian serum and formation of circulating immune complexes that contributes to the pathogenesis and autoimmune disorders, SLE, systemic lupus erythematosus or lupus nephritis and glomerulonephritis. These were like drug reactions, hepatitis and trypanosomiasis, malaria, etc. So when coming to the type 4 hypersensitivity reaction, this is also called as a delayed type of hypersensitivity reaction. It is a cell-mediated hypersensitivity reaction. It is commonly called as delayed type. It is only the class of hypersensitivity reaction to be triggered by the antigen-specific T cell. So, cell-mediated immunity reaction. This is mediated by the T cell dependent effector mechanisms that involve both the Th1 cells and cytotoxic T cells. So, the antibodies that did not play a role in type 4 hypersensitivity reaction. On activation, the Th1 cells that releases the cytokines which cause the accumulation and activation of macrophages, which is which in turn cause the local damage. So here the examples are tissue damage that occurs in bacterial like tuberculosis and leprosy, and viral measles, herpes, fungal candidiasis, and histoplasmosis, and parasitic leishmaniasis, sticho, infections like contact dermatitis from exposure to the chromates, and poison, insect bites, psoriasis, inflammation, and inflammatory effects in the bowel disease that leads to the production of Th1 type and Th2 like NKT reactions like ulcerative colitis to intestinal bacteria. A celiac disease is an aberrantly response in wheat gliadin. So this is called type 4 hypersensitivity, delayed type of cell mediated hypersensitivity reaction. When coming to the autoimmune disorders, as we discussed in the last classes about the sorry for the technical glitch. So the autoimmunity, which is Whitesby, the body is normally able to distinguish its own self antigens. So here from non-self antigens, and that does not mount an immunological attack against itself, so that this phenomena, this type of mechanism is known as immune tolerance. So here autoimmunity, it is an important development of a normal immune response. Apart from the major histocompatibility complex and idiotypic recognition, the immune system that does not normally react to the self. So here the broken down of self tolerance that leading to the immune autoimmune diseases. So there will be like 5 to 8% of individuals that develop autoimmune disease. More than 80% autoimmune diseases that have so far been identified, uh, which is affected only to a specific one tissue or one organ, while the others are the systematic autoimmune disorders. In organ specific autoimmune disorder, an immune response that is directed towards antigens in a single tissue or organ. The organ specific autoimmune diseases are Graves' disease, Hashimoto, thyroiditis, and type 1 diabetes. These the targeted autoantigens and lesions are restricted to a particular organ. In non-organ, like systematic autoimmune disease, those were like SLE, systemic lupus erythematosus, rheumatoid arthritis, and dermatomyositis, and autoantibodies that have widespread reactivity, and the lesions that involve the deposition of circulating immune complexes. So the immune uh, deficiencies, when the immunity, like immune deficiency diseases that were uh, described in the year of 1950s, the usage of antibiotics and the long-term usage of antibiotics and the passive immunization that increase the survival of the individuals with recurrent infections. The immune deficiency disease are characterized by the infections that are caused by the organisms, which are easily overcome and eliminated in healthy persons. In addition, the individuals with the immunodeficiency diseases that tend to contract infections very easily, that have aggressive disease and respond poorly to the therapy. So the immune deficiency diseases that are classified into two types. Those were like primary and acquired. So the primary immunodeficiency disease, which is causes the primary immunodeficiency disease, is the congenital due to the development and genetic abnormalities during the pregnancy. That may be genetic, which occur due to the inheritance of an abnormal gene from one or both parents. The primary immunodeficiency disease, which is a diverse group of disease that often presented with similar clinical signs and symptoms. They may be affect any of the cells that involved in immunity, or indeed chemicals such as complement components like T cell abnormalities, which include the George and Viscott Aldrich syndrome, and autoimmune lymphoproliferative syndrome, and X-linked hyper IgM syndrome, and autoimmune polyglandular syndrome type 1, and autoimmune polyendocrine crinopathy, candidiasis, ectodermal dystrophy, and X-linked gamma globulinoma enema, and chronic granulomatous. So the acquired immunodeficiency, all these are like in the primary immunodeficiency, like hyperimmunoglobulin syndrome and congenital neutropenia syndromes. All these were like a selective IgA deficiency that is common for the primary immunodeficiency diseases that appears to involve abnormalities in the switch recombination events. In the other hand, the acquired immunodeficiency disease called AIDS. In the recent years, the acquired immunodeficiency syndrome that is receiving more attention, but other acquired diseases that are becoming more common due to the use of chemo and radiotherapy. These are known as iatrogenic immunodeficiencies that is acquired as a result of treatment for another clinical conditions. The acquired immunodeficiency syndrome is caused by the infection with the human immunodeficiency virus, HIV, that is binding to the target cells that using the CD4 molecule, but the infection that requires the presence of co-receptors that includes the CXCR4 and CCR5, the chemokines receptors. HIV is a retrovirus containing a reverse transcriptase that allows proviral integration into the host genome. The HIV infection that leading to a range of immunological disorders that leading to overwhelming the infections and the tumors. For example, secondary immunodeficiency disorders like multiple myeloma, the cancer of the plasma cells, which produce antibodies, AIDS, immune complex diseases like viral hepatitis, and cancers of immune system like leukemia. So the other topic is vaccines. Here the vaccine is the term which originates from the Latin word called variolae vaccinae, cowpox. So here our scientist Edward Jenner is the one who first developed the first vaccine in the year of 1795, 1798 to prevent the smallpox in the humans. Today the term vaccine that applies to all the biological preparations that produced from the living organisms to enhance the immunity against the disease and either prevent or treat the disease. The traditional vaccines that containing the parts of microbes or whole microbes that have been killed or weakened so that they do not cause disease. When immune system that confronts these harmless versions of germs, it quickly clears them from the body. In other words, the vaccines that trigger the immune system, but at the same time, that teaches that this is the one which teaches to our body the important lesson about how to defeat its opponents. When inactivated or weakened disease causing microorganisms enter the body, they initiate an immune response. This immune response, which mimic the body's natural response to infection. But unlike disease causing organisms, vaccines, those are made up of the components that have limited ability to defeat its opponents. So, when inactivated or weakened, the disease causing microorganisms that enter, they initiate an immune response. This immune response that mimics the body's natural response to the infection. But unlike the disease causing organisms, vaccines that are made up of the components that have limited ability and completely unable to cause the disease. The vaccines, which are crucial to maintain the public health, they are safe, cost effective, and efficient way to prevent the diseases. The vaccines that led some greatest public health triumph. Uh, this can eradicate in a natural way that occurring the smallpox from the globe and the near eradication of the polio. The vaccine which provide the artificial acquired immunity that are easily less risk way to become the that can be classified as a non-toxic or non-virulent preparations of antigen material that can be used to enhance, induce, or long-term humoral as well as cell-mediated immunity response against the pathogen. The vaccine that are exposed either the entire disease causing microorganisms or some of the components that are that may be classified or constructed in a different ways. So from the components of the disease causing organisms such as specific proteins and the polysaccharides and nucleic acids, from living organisms that have been weakened usually from cultivation under the suboptimal conditions are called as attenuation, or from genetic modifications, the one which lead the effect of reducing their ability to causing the diseases. From whole organism that have been inactivated by chemical, thermal, or the others. From the linkage, the conjugation of the polysaccharides to the proteins, from inactivated toxins of toxin producing bacteria. Okay. Yeah, the immunization and childhood vaccination. Uh, the everyone knowing about this immunization process. So, when coming to the immunization, to the infectious microorganisms that can be achieved by active or passive immunization is such a way uh, which create the immunity that can be acquired by two processes: natural process that usually by transfer from the mother to fetus or by previous infection by the organism. The artificial process, which infection of antibodies or vaccines, the antigen used for inducing passage immunity with the microbial pathogens that induce the immunity but do not disease, or with the antigenic components from the pathogen. Passive immunization, as we know, here the passive immunization, the aim of passive immunization is transient protection or elevation of existing condition. The passive immunization is which the performed antibodies are transferred into the recipient occur naturally by transfer of maternal antibodies, development of the fetus. The maternal antibodies to mumps, diphtheria, rubella, etc. So here, in case of poliovirus and streptococcus, afford passively acquire for the reading of equally takes place. So here, this is a maternal antibody that can be achieved by injecting a recipient with proliferative antibody. So, in the past, before antibiotics, because of immunization that provides major diseases, despite the risk that has been occurred by injecting the animal serum, usually horse serum. This was first effectively um, therapy for otherwise fatal disease. So the when coming to the active immunization, the goal of active immunization is to elicit the immunity or immunological memory when active immunization is successful. A subsequent exposure to the animal elicits or antigen elicits that highlight the immune response that is successfully eliminated or eliminates the pathogen or prevent disease mediated by its products. So the active immunization that can be achieved by the natural killer cells, NK cells, and Nagara sir, and this one, despite the uh, in uh, passive immunization, in active immunization, the involvement of T and B cells that results in the formation of the memory cells. So, active immunization with the various types of the vaccines that have played the important role in the reduction of the death from infectious diseases, especially in the children when vaccination of children is began at about 2 months of age. So this is called active immunization. The major difference in between the passive immunization and active immunization is the transient protection or the elevation of the existing condition is passive immunization. So where the antibodies are transferred to the recipient, that occurs naturally by transferring of the maternal antibodies across the placenta to the developing fetus. Whereas in case of active immunization, the active immunization is to elicit the protective immunity and the immunological memory where the active immunization is successful, a subsequent exposure to the antigen elicit the immune response needs the pathogen or prevents the disease mediated by its products. So, the active immunization that can be achieved by the natural infections with the microorganisms, or it can be acquired the artificially by administration of the vaccine. In active immunization, the immune system that plays an active role in proliferation of antigen reactive T and B cells that results in the formation of memory cells. The active immunization with the various types of vaccines from the infectious diseases, especially among the children's, the vaccination that the process in the children is began at about 2 months of age. So, in the childhood vaccination, vaccination, uh, the schedule of the vaccination is important. It protect the infants and the children against the infectious agents that is prevented. The schedule indicates that where the vaccine or which vaccine are needed at what age, and how many doses that indicated by the members in the symbols. So, hepatitis B vaccine, it is given to mostly newborn babies before they are the second dosage that is given at the age of 1 to 2 months, which followed by the third dosage at the age of 6 to 18 days month 6 to 18 months, and the retrovirus vaccine, which is depending on the vaccine, uh, rotavirus, sorry, not retrovirus, which is depending upon the vaccine, which is used uh, 2 or 3 doses of the vaccine that are given at the ages of 2 months, 4 months, and 6 months. When coming to the Haemophilus influenza HIV vaccine, that is depending upon the vaccine used 3 or 4 dosages of HIV vaccine that is given at least at the age of 2, 4, 6, and 12th month and 15th month. So, the polio vaccine, the vaccine which is administered in as 4 doses of vaccine that are given at ages 2 months, 4 months, 6 months, and 18 months, and 4 to 6 years. So, the diphtheria, tetanus, and pertussis vaccine, before the age 7, the children are given the diphtheria, tetanus, and pertussis preparation. 5 doses of DTP are given at ages 2 months, 4 months, 6 months, 15 to 18 months, and 4 to 6 years age. The DTP, the 5 doses like that are given at of an and 7 to 12 years. For doses of vaccine are given at the ages of 2 months, 4 months, and 6 months. So, and 12 to 15 months, where the hospital people, they will provide a sheet which gives all these information when the baby is born. So the other vaccine, like pneumococcal vaccine, it is like 4 doses of vaccine that are given at ages 2 months, 4 months, 6 months, 12 months, and 15 months. So meningococcal vaccine, it is also having the 2 doses at the age of 12 and 16. It is not shown in the schedule sheet, but this is have to uh, 2 doses should have to take. So the other vaccine is influenza vaccine. The influenza vaccine should be given at the age of 6 months, and most of the people that need only 1 doses. The children who are in 6 months to 8 years old are given 2 doses at least 4 weeks apart if they are receiving the influenza vaccine from the first time. And MMR vaccine, measles, mumps, and rubella, 2 doses are given at the age of 12 to 15 months, and 4 to 6 years. The varicella vaccine, those like 2 doses, those were like 11 to 15 years, 1 doses, and 4 to 6 years. Hepatitis A, 2 doses of hepatitis A vaccine are needed to uh, take. Like the first doses is given at the age of 12 to 23 months, and the second doses is given to 6 to 18 months later. If the children over the age of 23 months, they have not been vaccinated, they can be given hepatitis A vaccine to help to protect from hepatitis A. And the other one is human papilloma vaccine. It is given to the adolescent girls and boys in 2 to 3 doses. Those given the first dose 9 to 14 years, that are given 2 doses separated by at least 5 months. Those given the first dose at the age of 15, that are given 3 doses. The second dose is given at least 1 month after the first, and the third doses is given at least 5 months after the first dose. So from birth to 11 to 12 years, if we include like meningococcal vaccine and the human papilloma virus vaccine, yes, definitely we have to take up to 16 years. So when coming to the types of vaccines, here there are different types of vaccines. We have mainly 4 main types of vaccines we discuss here is live attenuated vaccines, inactivated vaccines, a purified macromolecules as a vaccines, DNA vaccines. So the live attenuated vaccines, name itself indicating that it is attenuated vaccine which is live, used as a weakened form of the germ that cause a diseases. Because these vaccines are so similar to the uh, natural infection that they help strong and long lasting immunoresponse. The microorganism that are attenuated to lose their ability to cause the disease like pathogenicity, but retain their capacity for transient growth within the inoculated host. So, an attenuated strain of Mycobacterium bovis called BCG vaccine, a Bacillus calmette gorin. So, this Bacillus calmette gorin BCG was developed the Sabin polio vaccine and the measles vaccine, both consisting of the attenuated viral strains. So, here the examples for the uh, live attenuated vaccines are BCG vaccines, MMR vaccine, rotavirus vaccine, chickenpox vaccine. And inactivated, when coming to the inactivated vaccine, this inactivated vaccine, here the inactivation of the pathogens, these pathogens are the microbes. They can be inactivated by using the chemicals or heat is no longer capable of showing the replication in the host. It is important to maintain the structure of epitopes on surface of antigens during the inactivation. The inactivated viruses that use the killed version vaccines are used to protect against of the germ that causes a disease. Inactivated vaccines usually do not provide immunity that as a strong as live vaccines, so that may need several doses over time is called like a booster shots in order to get ongoing immunity against the diseases. So here is the inactivated polio vaccine or hepatitis A vaccine are the examples for the inactivated vaccines. When coming to the purified macromolecules as a vaccine, some of the risks that are associated with the attenuated or the killed, the whole organism vaccines that can be avoided with the vaccines that can consisting of the uh, specified or purified macromolecules. Those were derived from the pathogens. The general forms of such vaccines are inactivated like exotoxins and capsular polysaccharides that submit and conjugate form and recombinant microbial antigens. The subunit and conjugate vaccines, both the subunit and conjugated vaccines, those were contain only the pieces of pathogens, the parts of pathogens. They protect against to provoke a response from the immune system. For example, like pertussis vaccine and influenza vaccine. When coming to the conjugated vaccine, those were made using the pieces from pieces of coats of uh, bacteria. These coats are chemically linked to the carrier protein. The combination of as use a more powerful both HIV vaccine containing the polysaccharide that is joined to Tetanus toxoid, polysaccharides from the surface of the bacteria, which causes the pneumococcal disease that join to the diphtheria toxoid. So the other one is polysaccharide vaccines. Here the polysaccharide vaccines, those were the coating of capsule with antibodies or a complement that greatly increase the ability of macrophages and the neutrophils to the phagocytose, such as pathogens. The polysaccharides, here the polysaccharide are nothing but sugar molecule that are taken from the outside layer of the encapsulated bacteria, such as Streptococcus pneumonia, pneumococcal pneumonia, for the use of pneumovax vaccine. The vaccine for Neisseria meningitis, a common cause of bacterial meningitis, also consisting of purified capsular polysaccharide. When coming to the common toxoid vaccines, the toxoid vaccines use toxoid harmful product made up of by the germ that caused a disease. They create the immunity to the parts of the germ that cause disease, that instead of a germ itself, the immunity response is triggered to the toxins instead of the whole organism. Here, some bacteria, which include the cause of diphtheria and tetanus, produce the exotoxins. Uh, Here, exotoxins that is released from the gram positive bacteria, and these exotoxins that may produce many of the diseases, disease causing symptoms that results from the infection. Diphtheria and tetanus vaccine made by the purification of the bacterial exotoxins, then inactivating the toxin with the formaldehyde to form a toxoid. The vaccination in the toxoid that induce the anti-toxoid antibodies, which are also capable of binding to the toxin and neutralizing its effect. The tetanus, diphtheria, and pertussis toxins that are used to provide the vaccines. The recombinant vaccines are the genes that encode the major antigens of especially virulent pathogens that can be introduced to the attenuated virus and the bacteria. A number of microorganisms that have been used for vector vaccines, including vaccinia virus, the Coronipox virus, and attenuated polio vaccine, and adenovirus, and attenuated strains of Salmonella, the BCG strain of Mycobacterium bovis, and certain strains of Streptococcus, the normally existing the oral cavity. Our last topic is in the vaccines are types of vaccines are DNA vaccines. In the recent years, recently developed vaccination strategy is developing the DNA vaccines. Those were like plasmid DNA that encoding the antigenic proteins that are injected directly into the muscle of the patient or the muscle of the recipient, not the patient, recipient. The muscle cells take up the DNA, and the encoded protein antigen is expressed, leading to both humoral antibody response and a cell mediated response. Another investigational approach to vaccination that involves the introduction of genetic material that encoding the antigens against which an immune response, the body's own cell that can be used this genetic material to produce the antigens. The potential advantage of this approach that include the stimulation of a broad long-term immune response, the excellent vaccine stability, and a relative ease of large-scale vaccine manufacturation. The DNA vaccines to address several vital or several viral disease threats during the outbreaks, including SARS, coronavirus, the year of 2023, and a H1N1 avian influenza in 2005, and H1N1 a pandemic influenza in the year of 2009, and Zika virus in the year of 2016. So this is these are all the types of vaccine. So uh, the vaccine which originated from the Latin word variola vaccine. So the vaccine which provides the artificial acquired immunity, which is identified or which is developed by the scientist Edward Jenner in the year of 1798. As just we discussed, there will be a different types of schedule. Vaccination schedule is provided by the government these days, because it protect the infant and the children against the different types of the infections. So this is how the vaccines works. Thank you all.